Okay. Good day and welcome to our virtual forum presentation. I'm Angelo Victor Licaros and I will be moderating this event's proceedings. This forum is convened with a thematic focus centering on the stages of cellular division, as well as its pertinence to the field of cellular biology. The presentations and discussions that will commence today are held and made possible virtually amid the COVID-19 pandemic through the use of the Zoom online video communications platform. This event is hosted by students of Biology 150, Cell and Molecular Biology from the University of the Philippines, Manila, and held in fulfillment of the lecture requirements of the said subject. To help me in moderating this forum session, I have the assistance of two forum secretaries, Ms. Nicole Ira Manere and Ms. Patricia Claire Medina. Now, to formally begin these proceedings, I would like to call on Forum Secretary Nicole Manere to introduce the forum's forum topic as well as the overall goal of today's presentations.
The cell cycle has long been a pertinent concept in the field of biology, given its universality in different forms of life. While it is introduced early on in primary education, this forum aims to give the topic a more thorough, in-depth and organization.
Good day and welcome to our virtual forum presentation. I'm Angelo Victor Licaros and I will be moderating this event's proceedings. This forum is convened with a thematic focus centering on the stages of cellular division, as well as its pertinence to the field of cellular biology. The presentations and discussions that will commence today are held and made possible virtually amid the COVID-19 pandemic through the use of the Zoom online video communications platform. This event is hosted by students of Biology 150, Cell and Molecular Biology from the University of the Philippines, Manila, and is held in fulfillment of the lecture requirements of the said subject. To help me in moderating this forum session, I have the assistance of two forum secretaries, Ms. Nicole Ira Manere and Ms. Patricia Claire Medina. And now, to formally begin these proceedings, I would like to call on Forum Secretary Nicole Manere to introduce the forum topic as well as the overall goal of today's presentations.
The cell cycle has long been a pertinent concept in the field of biology, given its universality in different forms of life. While it is introduced early on in primary education, this forum aims to give the topic a more thorough, in-depth explanation to allow listeners to gain an appreciation of its complexity and organization. The discussion to ensue will be divided into 5 segments, putting emphasis on each specific stage of mitosis.
The overall flow of this forum's presentations will be as follows. There shall be 5 speakers, each of whom will be given 10 minutes to discuss their presentations. After each presentation, a short summary will be provided by one of the forum secretaries, followed by the collation and addressing of questions pertinent to the current discussion. After all speakers have presented, another brief summation of the forum's events will be provided, and then this forum will be formally adjourned. With that, I would now like to welcome our first speaker, Mr. R.J. Benedict Latina. The focus of his discussion will be on the mitotic prophase.
Let us head on to the first step in mitosis, which is prophase. But before we do so, it is important to know the current state of the cell during this period. Recall that there were a few steps that had occurred before mitosis. These were the G1, S, and G2 phases of interphase. These steps resulted in the duplication and replication of the centrosome and the chromatids. The centrosome is known as the microtubule organizing center and is integral to the formation of the mitotic spindle, which is responsible for pulling the chromatids apart. These duplicated chromatids are bonded together by cohesin, as it acts like a bridge that links these paired chromatids together. We can now proceed to mitosis, beginning with prophase. As we all know, prophase is a stage where chromatin condenses to become chromosomes. This occurs through the action of condensin, a 5-unit protein subunit that is related to cohesin in an ATP-dependent manner. Condensin functions by encircling the supercoiled loops of DNA. Together with special component proteins or SMCs or structural maintenance of chromosomes and non-SMC proteins, the loops of DNA fold back on themselves, forming an elongated antiparallel coil with domains at both N and C terminals. Because of this compaction, the chromosome appears to be rod-like in appearance. The extent of the compaction is seen in this slide. As you may notice, the condensin has introduced positive supercoils into the DNA loops, which resulted in a more compact fit. Now that we have settled one integral aspect of prophase, let us now leave the nucleus and move on to another salient feature of prophase, which is the breakdown of the cytoskeleton and the subsequent initiation of the mitotic spindle assembly. In the previous modules, we have discussed the existence and function of structural components of cells that facilitate movement of cellular components. Recall that one of the most important structures during mitosis is the mitotic spindle, since chromosomal segregation depends on this structure. The mitotic spindle is composed of an array of microtubules. Some microtubules of the spindle are attached to the sister chromatid pairs. These microtubules are called kinetochore microtubules, while other microtubules are called astral microtubules since they radiate outward from the poles. This slide shows both the kinetochore and astral microtubules. The first step in mitotic spindle formation is the radial appearance of microtubules around a centrosome. The minus ends of the microtubules link to the centrosome, while the plus ends are the ones that elongate. As seen in the slide, two locations where astral microtubules radially come from can be seen. The movement of microtubules is attributed to their dynamic instability, or the process that involves the shrinkage and elongation of microtubules around the cell. This thread milling action is due to their inherent instability caused by the phosphorylation of MCDK. The polar movement of these centrosomes is also exhibited during prophase and is attributed to the elongation of the microtubules that are attached to the centrosomes. Over time and with enough movement, the microtubules of the centrosome find and stabilize each other. These are called interpolar microtubules, and the centrosomes are now known as spindle poles. Now that we have tackled the events inside and outside the nucleus, it is necessary to also tackle another event that is characteristic in prophase. This is the dissolution of the nuclear envelope. We have discussed previously how the chromatids condense into chromosomes and how the mitotic spindle is formed, but we have not discussed the interactions between the two. This is where the dissolution of the nuclear envelope comes in. The nuclear envelope acts as a barrier between the nuclear components and the cytoplasm. During prophase, the nuclear envelope, which is composed of the nuclear pore, nuclear lamina, and nuclear membrane, are all disassembled by the disruption of the nucleoporin subcomplexes, depolymerization of the lamin filament, and the mechanical disruption of the membrane by cytoplasmic dynein motor molecules respectively. Even with all of the dissolution that has taken place, some organelles such as chloroplasts, mitochondria, peroxisomes, and lysosomes remain intact during mitosis. The Golgi complex remains intact during the cell cycle for plants, yeasts, and flies. For the mammalian Golgi complex, it is disassembled and incorporated into the endoplasmic reticulum, or it may split into two, one for each daughter cell. Shown in the slide is the unlinking of the Golgi complex from their ribbon-like appearance to clusters, then to isolated stacks, and finally to a haze of Golgi. Shown in this slide is an image of a cell undergoing prophase. As you can see, the red figures in the image are the chromatids. Also evident are the two asters, each consisting of a centrosome and their microtubules that emanate from the centrosome. Note that the microtubules span the entire length of the cell with notable interactions with the microtubules of the opposite aster. Thank you, and this is the end of my presentation.
Thank you for that presentation, Mr. Latina. I now call upon the Forum Secretary, Ms. Manere, to provide a summary of the first presentation.
To recap, prophase involves three events. First is the condensation of the paired chromatids to chromosomes by condensin. Happening simultaneously is the assembly of the mitotic spindle by spindle assembly factors, and the subsequent migration of the centrosomes to the poles through addition on the plus end of the microtubule. However, the polar movement will be left unfinished by the end of prophase. Finally, the last event is the dissolution of the nuclear envelope.
Thank you. Let's now move on to the question and answer portion for the first presentation. I have here a few questions raised regarding the mitotic prophase, which perhaps the first speaker could clarify. The first question here, you mentioned how mitotic spindle microtubules arise from the centrosomes present in the cell. The presence of centrosomes implies the examination of an animal cell. How is this process accounted for in plant and fungi cells since they lack the spindle-forming centrosomes?
It is indeed true that plants lack centrosomes. In plants, particularly in the phanerogams or seed plants, an abundance of microtubules have been detected around the nuclear envelope where they are nucleated rather than the poles. Once they have been nucleated, the microtubules are brought to each spindle pole by motor proteins. The motor protein has multiple heads, which attaches to different microtubules. In the absence of a centrosome, this facilitates spindle pole formation. Upon the breakdown of the nuclear envelope, the microtubules organize themselves into prophase spindles and with the help of gamma tubulin lead to the nucleation of microtubules. Additionally, there has been evidence that unlike centrosomes that act as spindle centers, which are focused only on a particular location, plants possess a centrosome cloud, or that the formation of spindle fibers are more dispersed than in animal cells. In fungi, spindle formation occurs intranuclear or within the nucleus. Spindle pole bodies, or an array of proteins that nucleate the microtubules, are found in the nuclear envelope. Since spindle formation occurs outwards, microtubules emanate from the spindle pole bodies found in the nuclear envelope towards the cell cortex, where they would continuously elongate.
Thank you for answering that first question. I have here another question which was raised. It says, what would account for the polarity of microtubules causing the association of growth to the plus end only, while minus ends remain associated to the centrosomes?
The polarity of microtubules is accounted for by the alpha and beta tubulin subunits that comprise the microtubule. It is worth noting that GDP attaches to both alpha and beta tubulin subunits. However, it is with the nature of GTP at each particular subunit which leads to the characteristic function of the subunit. The GTP that is bound to beta tubulin is commonly hydrolyzed to GDP. With this, depolymerization most likely occurs in the region of the microtubule with the beta-tubulin. This region is called the minus end. Alternatively, the opposite occurs for alpha-tubulin, which leads to the growth of the microtubule at that end, which is called the plus end since GTP and microtubules are added there.
Okay, so I thank the first speaker for his presentation and for addressing those questions, as well as the audience for raising these questions. Let's now move on to the second speaker, Ms. Angel Nicole Marie Vidal, for her discussion on the next phase of mitosis.
Okay, so after prophase comes metaphase. I will be highlighting the following events as we go through this stage: the breaking down of nuclear envelope, further condensation of chromosomes, the creation of kinetochores, and the function of kinetochore microtubules at this stage. The first thing that occurs in this step is the breaking down of the nuclear envelope that cases the chromosomes. This event is important to allow the microtubules to attach to the chromosomes. The nuclear membrane disintegration is caused by the changes occurring in the three compartments of the nuclear envelope, which are the nuclear lamina, nuclear pore complex, and the nuclear membrane. First, let's talk about the nuclear envelope. In recent studies, it was discovered that the nuclear envelope develops invagination as the cell enters prometaphase. This is where dynein comes in. Dynein are microtubule minus N directed motor proteins that are associated with the outer nuclear membrane. Activation of these proteins will initiate movement as they attach to the microtubules and pull the nuclear envelope regions towards the minus end, resulting in a deeper invagination. Other regions of the nuclear envelope will then stretch and become distorted, leading to the breaking of the nuclear membrane. The fragments of the membrane will then be segregated and removed along the area of the chromosomes. These fragments will later be reused by the daughter cells to reform the nuclear membrane. Next is the dismantling of the nuclear pore complex. The nuclear pore complex is responsible for the macromolecular transport between the cytoplasm and nucleus. During the prometaphase, these complexes are broken down and separated into subunits. As you can see in the image, CDK1 or cyclin-dependent kinase 1 and PLK1 polo-like kinase initiate multi-site phosphorylation on nucleoporine 53, which is the key binder of the nuclear pore complex. This results in the disintegration of the NPC. Lastly, the changes that occur in the nuclear lamina. The nuclear lamina consists of fibrous proteins and lamins that associate together to form a meshwork of filaments under the nuclear membrane. This provides the structural integrity of the nucleus. The breaking down of this layer occurs when it is depolymerized, where lamins are phosphorylated by the CDC2 or cell division control protein kinase. Phosphorylation results in the breaking down and separation of the lamin dimers back into individual subunits. At this stage, the chromosomes also become more condensed, as seen in this figure. This occurs to prevent tangling or breaking of the chromosomes when the spindle fibers move along as mitosis occurs. And while this happens, the nucleolus also disappears. Now that the nuclear envelope has completely broken down, the microtubules will now have access to the chromosomes. Three steps will then occur: recognition, attachment, and force. Before the microtubules can attach and segregate the chromosomes, the DNA should first be recognized by the chromosome segregation machinery. In most eukaryotes, centrosomes are monocentric, meaning they are restricted to a single and specific region in each chromosome. Without a functional centromere, kinetochores will not form on the DNA, which will cause the DNA segregation to fail. Once the location of the kinetochore is specified, its formation follows. The kinetochore is made of more than 100 proteins, which can be classified into three categories: inner kinetochore proteins, outer kinetochore proteins, and regulatory proteins. The inner kinetochore proteins are various proteins attached to the centromeric heterochromatin of the chromosome. These are responsible for the connection with chromosomal DNA, chromosomal interface, centromere replication, kinetochore assembly, and size determination. On the other hand, the outer kinetochore proteins, together with the fibrous corona, are the ones responsible for the attachment of the kinetochore with the microtubules for chromosome movement. This event is known as congression. These proteins are also responsible for the microtubule motor activities and signal transduction. Lastly, the regulatory proteins are responsible for monitoring the kinetochore. Now, for the chromosomes to move and be transported, the kinetochores together with the microtubules must generate force. To do that, the kinetochores must attach to the microtubules. Through this, the microtubules were able to move the kinetochore and the chromosome towards the spindle pole. Additional information: through lateral interactions, CENPE, a plus end directed kinesin, attaches to the plus end of the microtubules and moves the chromosomes from the area of the spindle poles to the middle of the spindle. On the other hand, dyneins, which are minus end proteins, move toward the minus end of the microtubules and contribute in the microtubule fiber movement, microtubule capture, kinetochore signaling, and spindle organization. The lateral interaction is converted into an end-on attachment. The connected microtubule is then officially called the kinetochore microtubule. The microtubules on the opposite poles then interact on the other side of the kinetochore. These microtubules from opposite poles then tug the chromosome back and forth for these to possess an equidistant position from both spindle poles. That's for pro-metaphase. Thank you.
I thank Ms. Vidal for her presentation, and I'd like again to hand us over to Forum Secretary Ms. Manere to provide us with a summation of the second presentation.
Metaphase is characterized by the disruption of the nuclear envelope in response to chemical reactions in three parts of the nucleus, namely the membrane, lamina, and pore complex. This allows for microtubules to reach the kinetochore of chromosomes via lateral interactions that make chromosome transport much more efficient. These interactions are then rapidly shifted into an end-on attachment, officially forming the kinetochore microtubules on both sides that pull the chromosomes until they are each at equidistant positions from the pole.
Thank you very much. So let's now move on to the question and answer portion for the second presentation. If you could go through the following questions raised by the audience for clarification. The first question is there a particular signal or event after prophase that would activate the dynein motor proteins which break down the nuclear envelope?
Based on studies, the nuclear disintegration mechanism is still hardly understood, and most likely the reason why the nuclear envelope is broken down is because that is how the cell cycle is programmed. But I can give you other reasons how dynein motor proteins are activated. According to studies, as early as the G2 phase or prophase, two proteins are recruited in the nuclear envelope. These are the centromere protein F or CENPF and bicaudal leak cargo adapter 2. These proteins are responsible for recruiting and activating dynein motor proteins for them to work and do their job, which was explained earlier.
Okay, so thank you for answering that first question. There is a second question here that says, in the formation of the kinetochore microtubules, Can the lateral binding of an unattached chromosome happen with other types of microtubules, or does this exclusively happen with kinetochore microtubules only?
Okay? No, chromosomes attach exclusively to kinetochore microtubules. As for the other microtubules, the polar microtubules do not attach to the chromosomes, but rather interact with the polar microtubules on the opposite pole, at the equator, for the symmetrical shape of the spindle fibers, while astral microtubules make sure that mitotic spindles are in the correct position.
Okay, so I'd like to thank the speaker and the audience for the second presentation and this very informative question and answer portion. I would now like to welcome our third speaker, Mr. Joseph Oliver Mayor, who will be discussing the next step of mitosis.
So here we proceed with probably the simplest phase of the cell cycle, but has an equally important role in normal cellular development along with the other stages of mitosis, which is the metaphase. The metaphase marks the event in mitosis where the sister chromatids are now aligned at the equator or middle of the cells. Here we can see an illustration in the upper left corner of what is going on during the metaphase, and below it is a photomicrograph of a plant cell during metaphase. In summary, the events that occur during metaphase are as follows. First, the centrosomes are now positioned at the opposite poles as seen as in the two images. Then, the chromosomes are now aligned at the equator, also known as the metaphase plate, as denoted by the red breaking line. This imaginary plane is equidistant from the two centrosome poles. Lastly, the sister chromatids are attached to the microtubules of the opposite poles at the kinetochore, creating tension on the chromosomes, keeping them in place during metaphase. During metaphase, we can see a highly organized arrangement of microtubules radiating from the centrosomes at both poles. These microtubules, or MTs, are classified into three groups based on its function and location. Here is a short recap of what we were mentioned in the previous presentations. On the left, we can see a diagram of the microtubules radiating from the centrosomes, where green represents the astral MTs, which can be seen radiating away from the spindle. The kinetochore MTs, seen here as blue, attached to the kinetochore of the sister chromatids, and the polar MTs here as seen as red lines. And here we can see that they are overlapping at their plus ends. Below this diagram is a micrograph of a metaphase spindle with centrosomes, spindle poles, chromosomes, and the MTs are defined. And we can clearly, okay. Next slide, please. Although the spindle might appear stationary during this step, this is not necessarily true in the molecular level, since the microtubules are at a dynamic state during this phase. Using fluorescently labeled tubulin subunits, scientists were able to observe that the microtubules at the spindle body, which are the kinetochore and the polar MTs, experience a constant flux of tubulin being added at the plus end of the MTs, and a loss at the minus end. These net gains of MTs at the plus ends is accompanied by a net loss of tubulins in the minus ends. Thus, the tubulins are experiencing a poleward flux, where the subunits added to the MTs move from the metaphase plate towards the pole. As you can see here in the image on the left, the darker tubulins added at the plus ends are gradually moving towards the poles, replacing the light tubulins constantly lost at the plus and minus ends. These entire processes create tensions on the chromosomes, hence they can actually be observed oscillating during metaphase. Another concept that is thought to power the movement of the chromosomes during metaphase is the opposing forces conducted by motor proteins. It is thought that the plus-directed motor proteins on the chromosome arms interact with overlap or astral MTs, generating an injection force which pushes the chromosomes toward the equatorial plane. On the other hand, minus-directed motor proteins interact with the kinetochore microtubules, which are thought to cause a pulling force to the chromosome, causing it to move towards the pole, opposing displacement caused by the plus end directed motor proteins. And that is all for this presentation, and thank you for listening.
Thank you, Mr. Mayor, for your discussion of the mitotic metaphase. I now call upon Forum Secretary Patricia Claire Medina for her summary of the third speaker's points.
To sum it up, metaphase is characterized by three events: the centrosomes being situated at the opposite poles of the cell, alignment of the chromosomes at the metaphase plate and their centromeres lying on the plate, and attachment of the kinetochores of sister chromatids to the kinetochore microtubules originating from the opposing poles. The microtubules projecting from the centrosomes are also classified into three, namely axial empties, kinetochore empties, and overlap empties. The dynamic state of empties is explained by the net addition and net loss of tubulin subunits. Meanwhile, the interaction between certain motor proteins with the empties allow for the movement of the chromosome.
Okay, thank you very much for that summation. Now, if we can move to proceed to some questions raised regarding the third presentation for the speaker, Mr. Mayor, to answer. I have here three questions. The first of which states, what function might the tension buildup of the kinetochore and overlap microtubules caused by polar tubulin flux serve?
Well, the tension buildup on the kinetochore due to the poleward tubulin flux in the empties helps in maintaining the integrity of the mitotic spindle as well as maintaining chromosome position at the equatorial plane. Also, albeit not having a role in chromosomal movement, the microfilament flux is of a necessity in mammalian cells in preventing erroneous chromosomal segregation since its primary role is in positioning.
Okay, I have here another question. It says, why is it necessary to provide an ejection force for the chromosomes using two opposing motor proteins rather than just using one minus n-directed protein in order to move it?
The necessity for two opposing motor proteins in providing ejection forces to the chromosomes during metaphase lies in the fact that most motor proteins are unidirectional, meaning that they can only allow movement of cellular structures toward a single direction. Hence, they are required to work in tandem so as to keep the chromosomes, in this case the sister chromatids, at their proper position prior to their separation during anaphase.
Okay, for our final question in this question and answer segment, it says, how do centrosomes end up on the opposite poles of the cell?
The movement of the two centrosomes towards the opposite pole in lieu to the formation of the mitotic spindle is due to the actions of motor proteins interacting with the cytoskeleton and empty fibers present in the cell cytoplasm. This movement is facilitated by two groups of motor proteins, dyneins, which role is to pull the centrosome towards to their minus n direction movement. And there is also the kinesins, specifically the EG5 motor proteins, which pushes the centrosomes away from each other as their plus n directed movement move apart the antiparallel or overlapping polar empties.
Okay, thank you, Mr. Mayor, for answering the raised questions. Let's now move on to the fourth speaker, Ms. Louise Claire Ortiz, for her discussion on the succeeding phase of mitosis.
Okay, so we are almost done with our in-depth journey through cellular mitosis, but it just isn't over yet. Okay, so after the alignment of the sister chromatids during metaphase, what goes next? If we were on the same page, then I'm assuming what's on your mind is also right. The next stage of mitosis is anaphase, wherein sister chromatids separate as they migrate towards the cell's poles. The separation initiates when the cohesin proteins attaching the sister chromatids together are cleaved. After anaphase, the sister chromatids are completely separated and become the chromosomes of the future daughter cells. In general, the process is as simple as that. But by digging a little deeper, we will be able to know that it is actually composed of two simultaneously or sequentially occurring subprocesses. These subprocesses are anaphase A and anaphase B, and later we will be exploring them one by one. So to start off, let us describe the general movement of the sister chromatids during anaphase. As stated earlier, the chromatids separate and migrate to opposite poles. But what leads this movement? Are these the centromeres or the sister chromatids? Just think about it briefly, and if your answer is the centromeres, then you are correct. In this GIF, we can see the straight paths taken by the centromeres as they move toward the poles, while the attached chromatids sway or bend in wave-like movements as they migrate to the ends. This suggests that the centromeres lead its attached chromatid to the pole, making the chromatid appear as though it is dragged by the centromere. The dragged motion of the chromatids are referred to as the ragdoll movements, and this movement occurs at a rate of 1 micron per minute. Although this mechanism of movement is commonly seen in almost all vertebrate and yeast cells, there are of course exemptions, such as in the spermatocytes of crane flies and the plant endosperm, where the chromatid arms may lead their own movement instead of them being dragged by the kinetochores. But the movement of the chromosomes is not just a smooth sailing ride. There are also forces within the cell that oppose their migration towards the spindle poles. These are called the polar ejection forces or polar winds, which must be overcome by the kinetochores or chromatid arms in kinetochore leading or arm leading type cells respectively. So let's say you were given a plant endosperm. Which of the two would have to overcome the polar winds? Is it the centromere or the chromatid arm? Since a plant endosperm undergoes arm-leading anaphase, the chromatid arm would have to undergo the polar ejection forces to complete the separation. So we have characterized the actual movement of the chromatids during anaphase, so we can now proceed to the discussion of the actual mechanisms that power it. As stated earlier, we have anaphase A and anaphase B that occur simultaneously or sequentially in most organisms. Let's begin our discussion about anaphase A. Anaphase A is the mechanism we are most familiar with, as it explains the shortening of the kinetochore microtubules, which in turn empowers the movement of the chromosomes toward the poles. Most of us thought that it's a polar movement of the kinetochores and it's constantly unidirectional, but this is not always the case. This graph in the screen, yes, this, by Stevens et al. Shows the change in distance of the kinetochore from the spindle pole as anaphase proceeds. We can clearly see that the graph is generally decreasing, but there are certain points in time, specifically just prior to the 200 and 400 second mark, where the distance of the kinetochore increased rather than decreased. These small fluctuations in distance is caused by the nature of the kinetochore microtubules to alternately grow and shorten within a certain period of time. At that brief period wherein the microtubule elongates, the kinetochore slightly becomes farther from the spindle pole, rather than closer to it. With this, we can say that the chromatids and kinetochores do not move towards the poles in a straight motion, but rather in back and forth movements. So this movement is called directional instability. And a quick brain tease for everyone before we move on to anaphase B. We're all aware that the microtubules shorten and enable the poleward movement of the sister chromatids. With this, we can expect the microtubules to also thicken as they shorten because of the tight packing of all the matter within the microtubule. But scientists were able to observe that there's no thickening at all. So how is that possible? This is because, unlike muscles that shorten by contraction, microtubules shorten through the disassembly of some of its tubulin units. There are two models that explain how force is generated to move the chromatids towards the poles. Let's briefly go over the two models. The first model on the left shows how the motor proteins at the kinetochores of the chromosomes use energy from ATP hydrolysis to pull the chromosome towards the pole. These proteins are also the ones responsible for the shortening of the kinetochore microtubules by a tubulin unit disassembly. On the other hand, the model at the right depicts how depolymerization of the tubulin units of the kinetochore microtubule pull its attached chromosome to the poles. In this model, the motor proteins at the kinetochores do not mainly facilitate the poleward movement of the chromatid, but rather keep its kinetochore attached to the microtubule to ensure a successful anaphase. We can see that tubulin unit disassembly occurs in either models of force generation, so we will now learn the three mechanisms of this. Firstly is the simple mechanism which occurs in yeast. The microtubules contain a plus and minus end, such that the plus end is attached to the kinetochore, while the minus end is attached to the pole. In a simple mechanism, the tubulin units and the plus end undergo disassembly, or simply an organized removal, while the minus end remains static or does not change throughout mitotic anaphase. Secondly is a dual mechanism that is observed in mitotic human cells. It is named as such because both the minus and plus ends undergo tubulin disassembly or removal to shorten the microtubule during anaphase. And lastly is the mechanism observed in crane fly spermatocytes, where the plus end gradually grows through disassembly or addition of tubulin units, while the minus end undergoes disassembly at a quicker rate. This enables minus end disassembly to overcome the gradual growth of the microtubule due to the plus end assembly, leading to the ultimate shortening of the microtubule. In this mechanism, the shortest form of the microtubule is only composed of the newly assembled tubulin units at the plus end, with no minus end tubulin units remaining. And moving forward, we have anaphase B, which is the elongation of the non-kinetochore microtubules, which enables the further separation of the sister chromatids. In anaphase B, a pulling force that acts directly on the poles, a pushing force generated from the sliding of the overlapping microtubules, and the lengthening at the plus end of polar microtubules all occur as the cell elongates in preparation for telophase. In figure A, it can be seen that the non-kinetochore microtubules remain static. Next slide, please. Yan. We can see here that the non-kinetochore microtubules remain static and do not undergo any length change during mitotic metaphase to anaphase A. The stability and constant length of the non-kinetochore microtubules during metaphase to anaphase A is maintained by the flux motors, or specifically kinesin 13, which are depicted in the diagram in pink. And at the onset of anaphase B, it can be observed that the flux motors, or the pink structures in the diagram, are now already gone. This signifies the inactivation or turning off of the flux motors, which initiates the entire process of anaphase B. Once the flux motors have been activated, empty crosslinkers, specifically kinesin 5 motors, and empty associated proteins or MAPs, which are shown as small blue circles on the diagram, act on the overlapping microtubule midzone and facilitate their partial breakage and loosening that will promote microtubule or MP sliding. Simultaneously, dynein motors or cortical pulling motors, which are the purple structures in the diagram, act on the astral microtubules attached to the spindle pole to enable it to move closer towards the cell cortex. In addition, plus end polymerization and minus end depolymerization in the interpolar microtubules further widens the gap between the two formerly attached sister chromatids. And comparing S1 and S2 figures B and C respectively, we can observe the significant change in the non-kinetochore microtubules, as well as the position of the spindle poles that led to the increased distance between the separated sister chromatids. That would be all for mitotic anaphase. Thank you.
Thank you, Ms. Ortiz, for your insightful discussion on the anaphase. Let's now hear a brief summation of the main points from the fourth presentation from Forum Secretary Ms. Medina.
To summarize this step of cell division, anaphase is characterized by the migration of liberated chromosomes towards the opposite poles of the cell. This event is composed of two subprocesses that can occur either simultaneously or sequentially, namely anaphase A and anaphase B. In anaphase A, kinetochore microtubules shorten and allow the movement of chromosomes toward the poles through directional instability. This shortening is possible through the disassembly of the tubulin units as seen in three different mechanisms in yeast cells, human cells, and grain-fly spermatocytes. Meanwhile, anaphase B is denoted by the separation of the sister chromatids through elongation of the spindle as promoted by certain actions by empty cross-linkers. Cortical pulling motors also simultaneously act on the astral empties to enable the spindle's movement towards the cell membrane.
Thank you very much for that summary. Before we move on to the next speaker, there are again some questions raised regarding this presentation, which perhaps the fourth speaker can address. Namely, first listed here, in the initiation of anaphase, what exactly is responsible for the separation of sister chromatids from attachment by a cohesin, and when does this occur?
Thank you for that, Mr. Licaros. Anaphase begins just right after the chromosomes have already undergone biorientation. During the transition from metaphase to anaphase, the spindle assembly checkpoint mainly monitors the process to ensure successful disjunction of the sister chromatids. Firstly, the checkpoint ensures that the kinetochores are properly attached to their respective microtubules. Secondly, the anaphase promoting complex or cyclosome degrades both cyclin B and, more importantly, securin. With the absence of securin, the separase enzyme becomes activated and can now cleave the cohesin molecules that keep the sister chromatids attached prior to anaphase.
Okay, so thank you for answering that first question. I have here just one more question that says, is directional instability of the chromatids and kinetochores also affected by polar ejection forces, or is it solely accounted for by microtubule growth and shortening?
That's a very good question. As a matter of fact, directional instability is also affected by the polar ejection forces in an indirect and less significant manner compared to the effects of microtubule dynamics. This is because these polar winds do not target the kinetochore, but rather the chromatid arms. Kay et al. Proved that the alteration in the distribution of the polar winds could change how the chromatids move as they separate. They also showed how the removal of the chromatid arms, where the polar ejection forces are directly exerted on, and leaving behind only the kinetochores, showed significant changes in overall chromosome behavior during anaphase.
Okay, so again, I thank you very much for your presentation and for answering those questions. And now let's move on to our fifth and final speaker, Ms. Eliza Nicole Jarin, for her presentation of the last phase of mitosis.
All right, so now let us proceed to the last step of cell division, the telophase. Here is a GIF of how we usually visualize telophase, and I am guessing that most, if not all of you, are familiar with this illustration. Now let us take a look at the nitty-gritty details of this final step of cell division. Here we have 4 major events happening during the telophase. We should keep in mind that these events happen in succeeding order. First is the reformation of the nuclear envelope, followed by the reassembly of the nuclear pore complex, then the reformation of the nuclear lamina, and lastly, the decondensation of chromosomes. Notice how the prefixes re and d have been used in the following events. These prefixes usually indicate an event happening again, or it might suggest going back to its previous state, which is kind of what happens in telophase. The cell reverts back to its interphase state, only this time there's 2 of them. Alright, so now let us focus on each event. First is the reformation of the nuclear envelope. So we all know that the nuclear envelope, also known as a nuclear membrane, is a 2-lipid bilayer membrane surrounding the nucleus, which is encasing the genetic material of the cell. Now let us take a look at this step by step. For the initial step, as you can see here, we have nuclear membrane vesicles. These vesicles are formed during the nuclear membrane breakdown to the surface of chromosomes, which happens during prophase. So you can consider these vesicles as remnants from the parent cell. So as you can see, there are materials here and here associated with these vesicles. These are the lamins and the integral membrane proteins of the inner nuclear membrane. And here we have the chromosomes. So what really happens during this step is that these nuclear membrane vesicles fuse and associate with the surface of these chromosomes right here. This interaction between the vesicles and the chromosomes are mediated by the lamins and integral membrane proteins, the ones I mentioned a while ago. So after that, the resulting structure appears like this, vesicles being bound to each chromosomes. After which, the vesicles start to fuse, thereby forming a double membrane around the chromosomes, which look like this. Now, after the reformation of the nuclear envelope, the reassembly of the nuclear pore complex proceeds. These nuclear pore complexes are the ones responsible for controlling the passage of macromolecules into and out of the nucleus. So, here's a brief overview of how nuclear pore complex reassembles during the telophase. This assembly is initiated by the binding of at least 2 classes of vesicles to a chromatin surface, which then fuse and flatten. Afterwards, an invagination of the inner and outer membrane occurs. This invagination proceeds until the membranes meet and fuse, creating a pore. Then, the pore is stabilized by the assembly of parts of the spoke ring complex. As the complex continues to reassemble, central materials are inserted simultaneously, accompanied with the buildup of the components of the rings. This is then followed by the addition of the peripheral filaments. Alright, so now that the nuclear pore complex has formed, let us take a look at the succeeding event, which is the reformation of the nuclear lamina. The nuclear lamina is a rigid and proteinaceous layer found underneath the inner nuclear membrane. It is usually involved with numerous nuclear activities such as DNA replication, RNA transcription, and chromatin organization. Now let us look at how this nuclear lamina reforms. So first, 2 lamin polypeptides known as type A and type B lamins interact with each other. This interaction forms alpha helical regions wound up each other, forming a 2-stranded alpha helical coiled structure. This is then followed by a head-to-tail association of multiple dimers. The resulting structure is a 2D linearly elongated polymer extended laterally by a side-by-side association of polymers underlying the nuclear envelope, the inner nuclear membrane to be specific. Alright, so before we proceed to the next major event, let us have a recap of the first 3 major events that successfully happens during telophase. So we know that the nuclear envelope reforms by the fusion of the nuclear membrane vesicles and chromosomes, after which the nuclear pore complex reassembles by the invagination of the outer and the inner nuclear membrane. Then, the nuclear lamina is formed under the nuclear envelope by the coiling of the alpha helical that extends laterally along the cell. Now it's time to look at the last major and probably most important event during the telophase. All right, so once the nuclear envelope has reformed, the nuclear pore complex start to pump in nuclear proteins. The influx of nuclear proteins inside the nucleus then causes it to expand, and the condensed mitotic chromosomes start to decondense back to their interphase state. As mentioned, the new nucleus is then able to expand via the selective import of nuclear proteins from the cytoplasm. Since nuclear localization signals are not cleaved from proteins that are imported to the nucleus, the same nuclear proteins that were released into the cytoplasm after disassembly of the nuclear envelope during prophase can be reimported into the new nuclei formed after mitosis. This is why the daughter cells formed during mitosis are the exact replicates of their parent cell. Furthermore, as the chromosomes decondense, the nucleolus reforms as well. Upon the reformation of the nucleolus, the transcription of rRNA then proceeds, completing the return from mitosis to an interphase nucleus. Now let us take a look at the summary of what happened during telophase. So two daughter nuclei form in the cell, the nuclear envelopes arise from fragments of parent cell envelope, the nucleoli reappear, the chromosomes become less condensed, and it is eventually the completion of mitosis. Alright, so now that we've discussed the steps of cell division, there is actually another event that happens after telophase, which renders the complete division of the cell. That is cytokinesis, also known as the cytoplasmic division of the cell. Cytokinesis occurs both in animals and plant cells. However, there are differences on how it proceeds. Let us discuss them individually. Let us first discuss the cytokinesis in animal cell. Alright, so for the animal cell, cytokinesis is initiated by the formation of the cleavage furrow on the cell surface. As you can see here, this is the cleavage furrow which resembles a deep invagination. This cleavage furrow starts to deepen and spread around the cell until it completely divides into two. After the initiation of the cleavage furrow, contraction and constriction proceeds. This is also known as abscission. The one responsible for this contraction and constriction and the accomplishment of cytokinesis in animal cells is the contractile ring, which is this one. Right? The contractile ring consists of actin, myosin, and regulatory proteins formed under the surface of the animal cell during the cell division. As the contractile ring proceeds in constricting the cell, it will eventually pitch into two. Right. Afterwards, this will be the resulting structure. Membrane insertion then takes place, wherein a new membrane is formed and inserted into the cell membrane, next to the contractile ring by the fusion of the intercellular vesicles. Now that we've discussed how cytokinesis takes place in animal cells, let us now take a look at how it proceeds in plant cells. So here is a diagram showing the succeeding events of cell division in plant cells. Let us discuss them one by one. For the first event here, during the G2 phase, cortical microtubules and actin filaments rearrange and encircle the cell. This rearrangement gives rise to the pre-prophase band. Alright, so the pre-prophase band here can be analogous to a blueprint indicating and predicting where the future cell division and formation of cell plates will eventually take place. Now, looking at our next event right here, alright, so as discussed a while ago, we know that during telophase, the chromosomes reach the opposite poles. Here we can notice this evident overlap of spindle microtubules. Alright, so this is known as the phragmoplast. The phragmoplast is unique in plant cells. It serves as a scaffold for cell plate assembly and subsequent new cell wall formation. So it is technically a guide where the cell plate should assemble. These Golgi-derived vesicles here then, carry various materials needed for the formation of the cell wall. They associate themselves with microtubules and aggregate in the equatorial region, forming the early cell plate. After which, as you can see here during the cytokinesis, the phragmoplast here, which acts as the scaffold, reforms at the periphery of the early cell plate. Golgi-derived vesicles continue to be recruited in these regions in order to extend the cell plate outward. Eventually, as you can see here in the last figure, so the membranes of the extending cell plate meets and fuses with the plasma membrane of the mother cell, completing the new cell wall. Here, it is also evident that the cortical array of interface microtubules is reformed and reestablished in both daughter cells. All right, so here is an electron microscope image and diagram of the cell plate formation happening in plant cells during cytokinesis, as I've discussed a while ago. So that will be all for my presentation, and thank you for listening.
I thank the fifth speaker for her in-depth discussion, which ends the phases of mitosis as covered in this forum. If I could again call our forum secretary, Ms. Medina, to provide us with the summation of our fifth and final presentation.
Telophase has four major events that take place sequentially. The reformation of the nuclear envelope step occurs with the fusion of the nuclear membrane vesicles that are fragments of the parent cell nuclear envelope and is mediated by lamins and integral membrane proteins. Meanwhile, the reassembly of the nuclear pore complex takes place through the invagination of the inner and outer membranes. The third event, which is the reformation of the nuclear lamina, is characterized by the interaction of two polypeptides to form an alpha helical coiled structure extending laterally along the cell. Lastly, the chromosomes decondense alongside the pumping of nuclear proteins inside the nucleus. After telophase, cytokinesis takes place. In animal cells, this process starts with the formation of the cleavage furrow, which eventually constricts through the contractile ring. On the other hand, cytokinesis in plant cells is characterized by the assembly of cell plates and the subsequent formation of cell walls.
Thank you very much again for that summary. So now, if time permits us, let's go to the question and answer portion of our final presentation. If the speaker, Ms. Jarin, could address just a few questions raised by the audience. The first says, where are the broken down components, such as polypeptides and nuclear proteins, of major structures mentioned, like the nuclear envelope and nuclear pore, stored in between prophase and telophase?
All right, so these broken down components are usually not stored. Rather, they are released into the cytoplasm after its disassembly. Then, in the last stage of cell division, which is the telophase, as I've discussed a while ago, they start to assemble again and form the major structures.
Thank you for answering that first question. Just another one here. The second one says, at what point in animal cell mitosis does the formation of the contractile ring complex actually occur?
All right, so the formation of the contractile ring occurs during cytokinesis, as I've discussed a while ago. However, its assembly is initiated after the onset of anaphase. Then, during the telophase, the cleavage furrow forms, followed by the constant contraction and constriction, which will eventually give rise to the contractile ring. That's it.
Okay, again, I thank Ms. Jarin as well as all the presenters in today's forum for their discussions. There are no more presentations to follow, and this juncture formally ends the forum itself. What remains is an overall summation of the forum's events and discussions for the benefit of the audience. With that, may I again call both our forum secretaries to provide a brief summation of their record of today's presentations.
This forum was an opportunity to further delve into the cell cycle, with much emphasis placed on mitosis. The discussion was divided into 5 sections to expound on the pertinent changes in each stage. Prophase is characterized by the condensation of chromosomes, mitotic spindle assembly, and nuclear envelope dissolution. This dissolution persists in prometaphase as a consequence of chemical reactions in several parts of the nucleus. This process is also characterized by microtubule attachment to kinetochores. In the metaphase stage, chromosomes are aligned to the centrally located metaphase plate through the joint effort of motor proteins and the 3 types of microtubules discussed. Anaphase can be divided into 2 sub-processes on the basis of the parts involved towards migrating chromosomes to opposite poles, namely anaphase A and anaphase B. Telophase is characterized by the reformation of several nuclear parts, such as its envelope, pore complex, and lamina, prior to the decondensation into chromatin. The process is often followed by cytokinesis, a process that divides the cell via different mechanisms in animals and plants respectively.
Each presenter was allowed to elaborate on their topic through a question and answer segment, and the common themes present in the points raised were mechanisms and signals leading to particular events, and the roles of specific cellular structures and processes. The questions asked on the prophase were related to microtubule polarity and alternative microtubule mechanisms in plants and fungi. The prometaphase speaker was asked to expound on dynein motor protein signaling and as well as kinetochore microtubule formation mechanisms. 3 points raised on metaphase were about the functions of kinetochore tension buildup, chromosomal ejection forces, and centromere positioning. Clarifications on anaphase tackled the initiation of sister chromatid separation and directional instability. Finally, the questions on telophase focused on the breakdown, component storage, and the formation of the contractile ring complex in animal cell mitosis.
And that concludes the proceedings of this event. Thank you very much to all of those who made this forum possible, the speakers, the secretaries, and the audience, for what was surely a thought-provoking and insightful forum discussion on the inner workings of the mitotic cell cycle. Again, I'm Angelo Victor Licaros, moderator, formally signing off. We wish a pleasant day to all, and we sincerely thank you again for coming.